Hello? Can you hear me?
We can.
Okay, fantastic. Thank you so much. Apologies for not being there in person. I know I'm missing out on a lot, but nevertheless, I'm thankful that I can at least join virtually. It's my greatest pleasure to introduce our first keynote speaker, Professor Joe Fetcho, this morning. I have known Joe ever since I was a graduate student, and though I never really got to work in his lab, he's had a phenomenal influence on my research, and I'm sure there are many here who would feel the same way. And I think it's important for the younger people in the audience to know the contributions that Joe has made to our field and how his work has enabled later. So in the early 1990s, when many of you may not have even been born, when calcium imaging using synthetic indicators was gaining traction, Joe was the first one who recognized the value of combining the transparency of zebrafish larvae with the ability to image neuronal populations in a vertebrate during behavior. I mean, this was really revolutionary, and the idea to be able to look deep into the brain of a vertebrate animal and see what the neurons were doing was truly, mind-boggling at that time. Joe's initial seminal studies showed the pattern of recruitment of spinal and hindbrain neurons during swimming and escape behavior in larval zebrafish. He also observational studies with photoablation of retrogradely filled identified neuronal populations so that causality could be established. This was the pre-GCAMP, pre-multiphoton, pre-mouse cranial window, pre-virtual reality era. And yet this early work from the Fetcho lab showed that the larval zebrafish is the next big neuroethological model system and a vertebrate to boot. And to this day, we are reaping the fruits of what Joe established all those many years ago. Then in the early 2000s, he had begun to employ transgenesis and combining transgenesis with electrophysiology and imaging to probe neural circuits underlying behavior. These and molecular characterization of spinal neurons that form the basis of future investigations into the spinal motor network. The Fetcho lab also showed how developmental timing influences intrinsic properties of neurons and how in turn that allows recruitment of motor neurons and spinal interneurons into various behaviors in a set of landmark studies. Several labs continue to follow up on these studies to understand ground rules of how circuits controlling early versus late behaviors are assembled and used. This body of work is also important because it brings home the point that many animals have to face the challenges of development with the challenges of function at the same time. You don't have any room to first build everything to perfection and them as you're building them. To quote Joe, not quite verbatim, but he says this often. He says, there's no evolution if you get eaten alive. He probably can say it much better. I mean, something to that effect. Once my postdoc advisor, Holly Klein, and I visited his lab to learn all things zebrafish. We were surprised when we got there to see some mice in cages. Later on, we got to know that it was for his pet python, which was there in the lab. And these mice were used as food for the python. So you know what Joe thinks about mice as lab organisms. Joe has always been generous, ready to teach techniques, share unpublished data, or spend time and give feedback. And it's not just me. I've heard from many in the field who benefited from his generosity. Many of his students and postdocs have gone on to become established PIs in this field, and many of them are, I'm sure, at this meeting. He's also an academic grandparent and probably even a great-grandparent. And then there is many of us who don't come in the lineage directly, but who would consider him as a mentor and a great inspiration. So for today, when I asked him his title, he said it's a mystery. He also asked me not to mention Daniel Nella, so I'm not going to mention Daniel Nella Joe. Thank you for the opportunity to share this with all of you. Take it away, Joe.
Is it working? You can hear me? Great. Well, thank you, Vatsala. That was very kind and generous. And I'm really honored to have the opportunity to speak to you today. I thank the organizers and I thank you all for coming, and I hope that you'll appreciate what I have to say by the end. It's really important to me to be here at this time in my life because my very first work was presented in Zebrafish, was presented in this auditorium at Cold Spring Harbor in the very early days of the Zebrafish meetings. And now I'm at the end of my career. Now, it turns out that actually the person who invited me to this meeting was David Shopik, and he wrote to me, and he wrote something to the effect that, Joe, it's really important that the young people hear what you have to say. Now, I interpret that as, Joe, it's really important that you talk to the young people before you're totally senile. Okay? And notice I said totally, which tells you where I stand now. I actually do really care about younger people. I think creativity in the sciences, not just in physics, which has a reputation for it, lies with the young people. It's often disguised in biology because the younger people are in these big labs, and so the old, mostly guys, are taking credit for their work. But I think young people are the most creative. Certainly my most creative work was done when I was quite young as an assistant professor. And I think that's for two reasons. First, young people are more fearless, and they're also less indoctrinated in the current dogma. And that allows them to just blow past the barriers that older people are reluctant to challenge. So my talk today will kind of be slanted toward younger people, although I hope there are some parts of it that are of value to the old geezers like me as well. And there are some of you out there that I know from those first Zebrafish meetings. I won't mention you. I'm doing you a favor. I won't report how senile you are either. Although I have a table here. I could show my table. So the next thing that happened in my interaction with David was I asked him, David, what should I talk about? And he said, well, Joe, talk about what matters. Well, that's some great guidance, right? Because what matters, as you can imagine, is highly individually dependent. And so it's kind of challenging to talk about what matters. But I listened to David, and so my talk is, the title that was mysterious is, What Matters Most About Zebrafish Work, to me at least. Now, if I'm going to talk about what matters about Zebrafish work, I thought it would be good to start with something that matters to everyone, to get off, hopefully, on a positive footing. And so the first thing that I want to say that matters is, of course, zebrafish. And zebrafish matter to hopefully most of the people in this room, but again, even that depends on the individual. Now, I study neural circuits and behavior, and I want to give you a sense for why it matters to me, for someone interested in neural circuits and behavior, by reading something to you. In the early days of the NIH Brain Initiative, there were meetings to try to determine what should be the focus of the Brain Initiative, and I was at some of those meetings, and at one we were asked to make a statement about what we think kind of matters for the Brain Initiative. And so I made a statement, and I want to read you that statement because I'm too old to memorize this whole thing. And I should say that this meeting was dominated by people who worked on mammals, mostly rats, mice, but primates and even humans, God forbid. And I call this kind of my Zebrafish manifesto for neurocircuits and behavior. And it goes like this. The problem with progress in understanding the brain is that we are largely mired in murky mammalian models. You can imagine that this didn't go over very well with the audience. We know what categories of information we need to understand how adaptive behavior emerges from brains. These include things like the activity at the single cell level of identified cells everywhere in the brain during behavior, the structural and functional connectivity that leads to the activity, and so on. We cannot yet get this everywhere in the brain at the single cell identified level during behavior in most vertebrates, including humans and other mammals. But, this is an important but, if our goal is to understand most of what vertebrate brains do, including our own, we can get that much sooner from non-mammals. That's because all vertebrates do largely the same things. They take in sensory information, internal and external, and use that and experience to produce appropriate coordinated motor responses, to navigate through the world, to find food, water, and shelter, to avoid becoming food, which used to be a bigger problem for humans than it is now, to interact and mate with other animals, to learn associations about what behavior leads to good things and what to bad, and adjust behavioral choices accordingly. They do all of this with almost all the same brain regions: hindbrain, spinal cord, cerebellum, midbrain tectum, called superior clicus in mammals, basal ganglia, and similar neuromodulatory systems, which are much the same across vertebrates. Evolution usually retains good solutions once established, and it solved the core behavioral problems in vertebrates long ago with most of the same brain regions we have. So the answer to what most of our brains do is present in every vertebrate. We should start by studying the one where we can get all the needed information most easily. This happens right now to be transparent young zebrafish, where we can image activity anywhere with cellular resolution, high speed, image structure and its dynamics, image synaptic dynamics, record electrophysiologically from any neuron anywhere, or a few people like Dave McLean and Minoru Koyama can do that. I happen to not be able to do that, although I used to do that back in the day. And we can perturb in very cell-specific ways and even get EM connectomes of the brain. Now, even if there were magical new tools were invented for mammals and humans, we still would be better off first figuring out a brain with 100,000 neurons that does much of what all vertebrates do before trying millions or billions. We would then reveal the principles of brain organization and understand shared brain regions and behaviors, and we can clean up the residual variation like cerebral cortex later on. So I urge you to move out of models that are opaque in most respects. The path to understanding what most brains do, including our own, is clear, literally clear, and it runs through young zebrafish. Now, I realized the essence of this in the early 1990s when we started in vivo imaging activity in zebrafish, and my view hasn't changed. So zebrafish matter, even for me. What else matters? Now, this is where it gets more difficult, because zebrafish was an easy one, right? But as I get to other things that matter, my credibility matters too, because if you're going to listen to what I have to say, I have to be a credible source. And Václav did a good part to try to establish that I might have some credibility. But I've known Václav for a long time, and so you might think that she's not an objective source. So I decided that I would look into a more objective evaluation of my credentials. And so I consulted kind of the oracle of our age and our future overlord, ChatGPT. And I asked, what are Joe Fetcho's contributions to zebrafish neurobiology? And ChatGPT says, I apologize, but I couldn't find any specific information about Joseph Fetcho's contribution to zebrafish neurobiology in my training data, which goes up until September 2021. It's possible that Joseph Fetcho has made contributions in the field after that date. Not very many after that date, by the way. Or his work may not be widely documented in publicly available sources, if you have more specific information. Now, this is obviously somewhat troubling at this stage of my career. And as it turns out, I have done some things in my career, starting in the first publication on zebrafish was in 1995. And so I think I've done some things that matter in spite of ChatGPT. But I want to say something about the origin of zebrafish neurobiology, because I know there's some confusion among some people about the origins of zebrafish neurobiology, and I think we all need to be on the same page about this. And so what I was going to say, and you have to listen to this whole sentence before you get offended, anyone, okay? I have a tendency to offend some people, so let me just say that no one in this room established zebrafish as a model system in neurobiology. Unless they're one of these people. And Ruth, and I prepared this before I knew that Judith Eisen was here, and Judith's here somewhere, hopefully in this room, up there. So zebrafish neurobiology was established at the University of Oregon. George Streisinger had the vision to realize that zebrafish would be a great model to combine genetics with a transparent organism to study aspects of development, and he ultimately wanted to study behavior as well. He didn't really himself start the neurobiology side. That was these other folks. I guess I should use the pointer. That was these other three folks. These are the people who really put zebrafish on the neurobiological map, if you will. And that was happening when I was a graduate student. And they presented a lot of that work at the Society for Neuroscience meetings. And those are some of the most amazing experiments I've seen in my career. They involve things like Judith did, watching neuronal growth cones grow out in a live intact animal to see how they did their pathfinding to their targets. Or only Judith, I think, can do this, pluck out a motor neuron and move it from one place to another in the embryo and see if the location of the neuron changes the pathfinding of the cell. Monty Westerfield did some early work on behavioral stuff, particularly looking at motor neurons in somewhat older fish to ask how motor neurons were recruited during behavior in zebrafish. So if you don't know about this work, then find it and read it. You should know about it. It matters. Now, and this brings me to the next thing that matters: history and related things, ideas, and curiosity. So history matters. We need to respect the contributions of the people who came before us. And the way you do that is by reading the literature and knowing who did what before. That prevents you from just copying what they did, although people still do that. It's kind of lame, but they do it. And but you should know that stuff inside out. This is especially for the young people. Learn it, read it, and learn it while you still have a memory. And don't be ChatGPT. And there's another advantage of knowing the history, and that is people often ask me, well, Joe, do you have any ideas about what I might do? And the answer is read the damn literature and find out what the frontier is in the things you're interested in, and look at what the gaps are and what the major outstanding questions are, and use that to try to figure out what you're going to do next. It's kind of an obvious thing, but when I was doing that from early on in my career, as you'll see as I go on with my talk. And the final related thing is, for most of us, we got into this business because we were curious about biology, about how biological things worked. And we wanted to satisfy that curiosity. And there's different ways of satisfying that curiosity. The easiest way is to read the literature. If you want to know how something works, read the literature and see if they've already figured it out. And you can often find in the literature, and to me, it turns out to be almost as satisfying to read the literature as it is to do the experiments myself. And if it's not there in the literature, then you can always do the experiments. But it's a lot more time-consuming. So it's much nicer to just have someone have done it before and get the answer by looking at the literature. And you'll see how much the literature can matter in getting answers to questions as I go on with my talk. Now the next thing that matters is probably the most important one in my talk today, and it's one that maybe it's only second to my very last thing that matters. So stick with me. Oh, by the way, my talk isn't going to be 35 minutes. If you saw that slide that went past that said 35 minutes, yeah, I don't pay any attention to times. I'm too old for that, okay? And I didn't sleep at all last night, so it could be the end of the meeting. I'll be done by the end of the meeting, okay? I promise. So the next one is the really important one that matters to me. It's principles with predictive power. This is kind of a philosophical thing, philosophy of science thing. I have a long-standing interest in philosophy of science. I'm by no means an expert in it, but I took a couple undergraduate courses in philosophy of science, and I got kind of interested in it. And back in that day, which is in the late 70s, again, most of you weren't born, a large fraction of you probably. Back then, the focus was primarily on two aspects of philosophy of science, or roles of science. One was explanation, and the other was prediction. And this just shows you some of the, this just shows some statements from various philosophers of science over the years about the roles for prediction and explanation are the two main functions of scientific knowledge. Here, two principal aims, prediction and explanation, and so on. So prediction and explanation were back then the main focus, but it's starting to shift a little bit now, partly because of people like Heather Douglas, who is now focusing on prediction. And so I'm going to voice my own view off on you. I think that the most important thing about science and what sets science apart from other ways of coping with the external world is that science has huge predictive value. In fact, I'm going to really go out on a limb and say, I think the overall goal of science is to make the shortest list of the simplest principles that predict as much about the natural world, both physical and biological, as possible. Now, that's a very powerful thing to be able to predict things about the natural world. And it's especially powerful even in an evolutionary sense, because you can just give the list to your progeny or to your people in your society, and they can use it and you arm them with predictive power, which matters a lot. So you can use the principles of genetics to make predictions about how biological variability arises and to understand the mechanisms by which Darwinian principles of evolution might be happening. So principles with huge predictive power are key. So what does that have to do with brains and behavior? So let me introduce that with this slide. This is a phylogeny of vertebrate brains. Now the thing is, the brains and behavior is a huge problem. And it's a huge problem because there are a lot of brains. And there are a lot of animals. And each of these brains produces a variety of different behaviors, and different brains produce different behaviors. So we can ask the question, what does it even mean to understand the neuronal basis of behavior? Or another way to put it is, when you're asking questions like this, is when will you actually be done understand the neuronal basis of behavior? And I would submit that being done doesn't demand that we figure out the neural circuit for every behavior in every one of these animals, which certainly the NIH isn't going to fund. And instead, the hope is that there are principles of assembly and function that allow us to make predictions about how different brain regions are organized to produce behavior and how brains across vertebrates and other animals are organized. And my work on zebrafish has largely been directed toward trying to understand some of those principles of organization. I didn't start that way. I started with a much simpler goal. At the time I wanted to do a postdoc, there weren't really any circuits that were really well understood at the cellular level. We didn't have wiring diagrams of circuits for very many things back then. And so I set kind of a simple goal, relatively simple goal. It wasn't that simple, but relatively simple goal, which was to try to understand one vertebrate motor circuit from sensory input to motor output before I died. I only care about things that happen before I die, because afterwards it's not So important. And so I set out to do that for the escape behavior in goldfish. And I did it by using sharp intracellular recordings, which is the hardest way to do electrophysiology, if you don't know anything about electrophysiology. And we made a lot of headway in that. But I realized along the way that the future prospects of this were dim. And the reason is that most behaviors in vertebrates don't involve relatively few neurons. They involve whole populations of neurons. And we needed ways to be able to look at the activity in those populations of neurons and how it changes during behavior. And it happens that when I was in my first assistant professor's position in the early 1990s, I was interacting with a postdoc in another lab named Don O'Malley, who was doing calcium imaging in cultured neurons with confocal microscopy. And in talking with Don, I realized that with calcium indicators, and Václav alluded to this, we would be able to monitor activity of neurons with calcium indicators, whole populations of neurons in vivo in a vertebrate, which hadn't been done before and now is kind of the norm. And I just want to show you, I dug through my museum of former Apple computers, and I dug up some old movies of videotapes we made. We had to do videotapes back then because the speed of storage was so slow you couldn't stream them to a drive or anything, and drive capacities were nearly nil. So, and I just want to show you a couple examples from the early 1990s. These are some of the very first in vivo imaging of neuronal activity with calcium indicators. Some of them are not. This one in particular is not earth-shatteringly impressive, but these are motor neurons in spinal cord filled with a calcium green dextran by backfilling them from muscle. And what we're going to do is we're going to stimulate the body and get the fish to try to move. And you'll hear a sound when the stimulus occurs, a kind of train of stimuli. And you'll see the cells light up. It looks like they're getting bigger right there because the edges are dim, and when they get brighter, the outline of the cell gets bigger. So again, this is some of the very first imaging. This is in spinal cord. We got better at it than this, but we also did it in the brain itself. And this is the famed Malmner cell that does the escape response in zebrafish. And so let me show you that. And so we're going to tap the fish gently on the head. There's the response. It's kind of a piezoelectric sledgehammer. So it's probably, we were doing traumatic brain injury experiments on and so we had this, we did this stuff. It was enormously exciting at the time. I don't have the audio of the video tapes, but you can hear us yelling when we got our first responses on some of the videotapes. And so then the question becomes, well, what are you going to do? Okay, so you can watch cells light up. What the hell are you going to do with it, right? And at the time, if you look at the literature, and I knew this very well, that there weren't many principles of motor control, and my interest was in neural control of movements. But there was like one thing that was called a principle of motor control. It was Henneman's size principle, which some of you probably heard about. They still teach it in medical schools. And it basically refers to how motor neurons are recruited as the force of output from a muscle increases. Motor neurons with small cell bodies that innervate fewer muscle fibers and slower muscle fibers are recruited first. And then as force increases, you recruit bigger and bigger motor neurons with larger cell bodies that innervate many more and larger and faster muscle fibers. We had some information about from motor neurons, just from intracellular recording of motor neurons, mostly from cats actually back then. But we knew nothing about populations of interneurons because you just couldn't do the experiment. You couldn't record from groups of interneurons as populations of identified cells. And so the first thing we set out to do was try to ask the question of how are populations of interneurons recruited during behavior. And the people who did that work, some of the people who did that work are shown on this slide. It was really spearheaded by Dave McLean, who was a postdoc in the lab at the time, although other people made hugely important contributions too. And Dave figured out how spinal interneurons are recruited behavior and found that there were some similarities to the size principle, but also some differences and some extensions of ideas related to size principle in the spinal cord. We went on, as Václav said, to look at it in hindbrain as well. And the people who did the hindbrain work, and I'll be highlighting these people as I go through some of this stuff, are here. The main people here are Emina and Minoru. And other people were absolutely critical. Of course, Shinichi is one of them. Many even know of Shinichi, who's basically a transgenic genius. Actually, he's a general genius in my view. And some of his progeny who are equally talented. And so what I'm going to do is tell you about some of that work and some of the principles that I think emerge from that work. And then I'll try to extend that beyond zebrafish and even beyond vertebrates and give you a sense of whether these principles apply to as large a span of animals as possible, because those are real principles. Right? You want a principle that has explanatory power across all of phylogeny would be ideal, right? That's a powerful principle. Okay, so let me walk you through some of the hindbrain work, which some of you probably aren't aware of if you don't work on neurocircuits behavior particularly. So if you look at a zebrafish hindbrain in cross-section early in life at 5 days when most of the core behaviors are already being produced, the neurons are jam-packed up in the upper part of the hindbrain here, and down here is a neuropil. It's very invertebrate-like in a way. The connections are down here in this neuropil, and the cell bodies are up here, and the neurons send processes down, and they form connections down in the neuropil. And it looks fairly homogeneous, but it's not, thanks to the wizardry of Shinichi and his cohort. We have these transgenic lines that allow us to look at the organization of these neurons in live intact zebrafish. And this is a dual transgenic line that was made in by Shinichi and company, Shinichi and Co. These are glycinergic neurons in green and glutamatergic neurons in red. And they look kind of jumbled up. This is the spinal cord side, but this is a 3D reconstruction in which the hindbrain is labeled as well. And so I'm going to rotate it around, and you'll see that though they're jumbled in spinal cord, when you look in hindbrain, they're actually in columns. So there's segregated columns of glutamatergic neurons and interleaf columns of glutamatergic neurons and glycinergic neurons, glutamatergic and glycinergic neurons. And so we set out to figure out what the significance of these columns was, because in biology, when you have order, usually it matters for some reason or another. And so we set out to try to figure this out. And it turns out it's related to developmental processes involving transcription factors. And there's a transcription factor code that defines the generation of cell types in spinal cord. Progenitor cells in the spinal cord are occupy different zones, and these zones produce transcription factors that induce other transcription factors that in turn induce other transcription factors, so that neurons arise from these zones from ventral spinal cord to dorsal spinal cord, first really shown by Briscoe and Erickson, and Tom Jessel was involved to some extent too. This code, this transcription factor code, is highly conserved among vertebrates. So if you look at a couple of these transcription factors, it was first worked out, I should say, in mice and chick. And if you look at these two transcription factors in zebrafish, again, Higashijima lines, the CHOX10 neurons are more ventral in spinal cord, and these LHX2 neurons are right at the dorsal side of the spinal cord. And we can take a fish with these dual labelings and look at it at different places along the body axis, from the spinal cord here to the hindbrain. And what you'll see is that these patterns persist, although the topology changes. Here they're kind of horizontal, but by the time we get at the hindbrain, they're kind of more vertically oriented, more with the orientation of the transmitter columns I showed you earlier. And it turns out that these transcription factors are overlapping with some of the neurotransmitter columns. And we could show that in dual transgenic lines, and I'll show you one example of this, in which the transcription factors are going to be labeled in green, and the transmitter phenotype is going to be labeled in red. So let me turn this on. This is a cross-section of hindbrain. The midline's kind of right here. So this is the transcription factor. It's a very medial transcription factor. It's a CHOX10 transcription factor. And you'll see, importantly, this extends throughout the hindbrain. These are the otoliths in the ear. There are lots of circuits in hindbrain that do jaw movements and eye movements and body movements, suggesting that all these circuits are arising from a simple early patterning. So this transcription factor column, if we turn on the glutamatergic stripe labeling in red, this transcription factor overlaps glutamatergic neurons in this most medial glutamatergic column. So as someone interested in circuits, I was more concerned about how the columns differ from one another beyond differences in transmitter phenotypes. It might give us an idea of what those neurons are doing in behavior. So one of the first questions is, are the cells in different columns morphologically different from one another? And we explored that by doing experiments where we randomly labeled neurons in hindbrain with membrane-targeted fluorescent protein so we could then reconstruct them in 3D. And we labeled them in lines with the transcription factor or the transmitter phenotype labeled in a different color so we could figure out which column, which transmitter column the neurons are in and look at their morphology as well. And then what we did was we just sorted the neurons by what column they were in and asked how do the neurons in the same column compare to one another and how do they compare to neurons in another column. And this is where it really got kind of tantalizingly interesting. So let's look at what Mimina really was the person who did a lot of this work. This is the most medial glutamatergic column. What do the neurons look like in that column? Now these are, in this work, we're agnostic of what circuits these are in. We're taking any neurons in hindbrain that get labeled, and we're just sorting them by column. We don't know what circuits they're in. We don't really care at this point. And what you find in this case is that these neurons, we're looking down on the fish from above. The front of the fish, this is rostrum, this is called the front of the fish is up here, and the back of the fish is down here. These neurons, of course, are glutamatergic, and they all have a descending axon, and that axon stays on the same side of the body. So they're kind of, the jargon is ipsilateral descending excitatory neurons. Some of them have ascending processes, but what they share is a descending axon. What about this column here, this glycinergic column? Those are ipsilateral ascending glycinergic neurons. And what about the next glycinergic column over there, that one? Those are commissural glycinergic neurons that have outputs opposite the cell body, but no apparent axonal outputs on the same side of the cell body. And finally, the most lateral glycinergic column are bilaterally projecting glycinergic neurons. So this is quite remarkable. It basically indicates that these columns, in a way, are representing circuit components. And the implication of that is circuits in hindbrain are formed by drawing from a set of components produced during development. And we wanted to confirm that by asking how a network is built from these components. And the network we focused on was the network for escape behavior that zebrafish use to avoid attacks from predators. So this is a dragonfly, that's the zebrafish there, the poor, helpless zebrafish. Not so helpless. The dragonfly's attacking. The zebrafish does this escape behavior initiated by the Malner cell, which is this giant neuron in hindbrain that I showed you before in calcium imaging. And we knew a lot about the circuitry of the Malner cell from the work that I did my postdoc in Don Faber's lab. And a lot of the circuitry in hindbrain had been figured out. But Minoru Koyama in the lab set out to ask the question of how the neurons in the escape circuit in zebrafish mapped onto this columnar organization, expecting that they should map onto it in orderly ways based on the morphology and transmitter phenotype of the cells. This brings me to another thing that matters. The goal here in your experiments is definitive, enduring results. No half-assed jobs, okay? And the tools you use matter. The idea is to use the most definitive tool to reach your conclusion, and it shouldn't matter to you how hard it is. Just buckle up, Buttercup, and do the experiments in a way that gets you a definitive answer, okay? And so how do you do that in this case? Well, what you do is, and Minoru did this, is you go in and you record from the neurons and you ask, do they have the appropriate patterns of connectivity to be the neuron in the circuit. Basically, you do the same thing you do when you're actually trying to figure out a neural circuit for a behavior. You need to know who connects to whom, and is it excitatory, inhibitory, and how strong is that connection. And Monroe did this, and I'm just going to show you one example for this type of cell. This is a commissural inhibitory neuron, a glycinergic neuron, that gets input from the auditory nerve, which also has input to the Maunder cell, and then crosses over and inhibits Maunder cell on the other side, and inhibits the ipsilateral Maunder cell as well. And so what Monroe did is he went into the zebrafish and found these cells and did simultaneous recordings from the interneuron and the Maunder cell. And so the expectation then is that if we stimulate the auditory nerve, we should see excitatory input to the Maunder cell and excitatory input to the inhibitory neuron. Well, and these are his recordings. This is the inhibitory neuron being activated by a stimulus to the auditory nerve, and this is the excitation of the Maunder cell. And then the expectation is if this is the right cell, if we fire this cell, we should see inhibition of the Maunder cell, and that's what he did. And lo and behold, we fire the cell and we get inhibition of the Maunder cell. And then, to be confident that we have the right cell and that there's likely to be synaptic connections that he's, of course, directly shown electrophysiologically, he fills both of them with dye in different colors and reconstructs them in 3D in a line with the transmitter columns labeled so that he can map them to particular columns. And so here is that pair of cells. Here's the Maunder cell, here's the inhibitory neuron, and reconstructed in 3D, and you'll see that it has outputs on both sides of the body. It's a glycinergic neuron with output. He did pharmacology too to prove that it's a glycinergic neuron. So it's a glycinergic neuron with output on both sides of the body. So where would we predict that it would occur, given the patterning of these columns? Well, the glycinergic neurons with outputs on both sides are the most lateral of those glycinergic neurons. So it should be that column. And indeed, these are the hindbrain columns here, and it falls right at the bottom of this most lateral hindbrain column. And of course, if you want to do it definitively, you don't just do it once. So Monroe did it a whole bunch of times, and they all fell right at the bottom of this lateral column. They happened to fall near the dendrite, one of the dendrites of the Maunder cell. So he not only did this a bunch of times, kind of Herculean, unless you're Monroe, and then it's kind of an afternoon. I'm not kidding, he's incredible. And so he not only did that for that cell type, he did it for every cell type known in the escape circuit in hindbrain. And they all mapped as expected onto the expected positions in the columns in hindbrain. And these are some of the cells, morphology of some of the actual cells that he labeled in his experiments. So it was totally consistent with the idea that the way you're building circuits is by drawing components from columns of cells in hindbrain. But it raises the obvious question of all of these cells are at the very bottom of the column. What's the significance of this axis of the column? And so we explored that, and it turns out that this axis of the column represents the birth date of the neurons. The neurons that were born first and are oldest are at the bottom of the column, and the youngest, most recently born neurons are up here. And we know that from color change experiments, which express Kaeda in a line that Shinichi generated, of course. And you take a fish in the egg, it's expressing this Kaeda. Some of the neurons in that have differentiated in that column, and they're green. But if you shine a UV light on it, you can convert them to red. And then if you wait and let them grow up, new neurons will be generated. They'll make fresh Kaeda. That will be green. And then you could just look at the fish afterwards and see which neurons were around at the time you converted it. Most of you know about these Kaeda techniques, but some may not. So, and what you find is that the old ones, the ones that were converted before, are in red, and the newer ones that were born after the conversion are in green. And that pattern seems to extend through the hindbrain. There's some distortion here, which I'll return to a little bit later on. Old at the bottom, young at the top. Now, what else varies along that axis? The morphology of neurons also varies along that axis. If we just ask how extensive the axonal arbor of a neuron as a function of position along this axis, there's a nice orderly relationship. So this is a plot of the bottom of the column to the top of the column, and the axonal, total length of axonal branching traced in 3D for neurons at different locations from the bottom to the top of the column. You'll see that there's a nice relationship. The neurons near the bottom, the oldest ones, have the longest axonal arbor branching, and the youngest ones have shorter axonal arbor branching. And that's not just because the youngest ones are younger and haven't had time to grow, because we can watch how fast they grow, and these ones are growing more slowly from the beginning. So it's not just that these have more time to grow. So there's a difference in axonal branching. But what I really was most interested in is what are possible functional differences between these neurons? And we knew that some of these neurons were involved in swimming behavior because Minoru recorded from some of these neurons in hindbrain and could show that they're rhythmically active during swimming. Now, fish can swim over a large range of speeds. They spend much of the time swimming slowly, kind of beating their pectoral fins, moving at about a body length per second, maybe 15 to 20 hertz tail beat frequency. But if you poke them, they do an escape response, which they can move at 40 hertz, 40 body lengths a second, and over 100 hertz in some cases, tail beat frequency. So we asked the question was about which neurons in the column are involved in different speeds of swimming. And we did that with calcium imaging. We asked, what's the minimum swimming frequency at which a neuron in the column lights up during swimming, and how does that relate to its position from the bottom of the column to the top? And so this is the minimum swimming frequency at which a neuron lights up. And what we found is that the neurons that light up at the lowest swimming frequencies are up here, and the neurons that light up when the fish is going faster are down here. So there's kind of a speed map where these are the neurons that are doing the fast, biggest body movements, and these are the ones that are doing the slower movements. And in fact, the fastest motor behavior the fish produces is the escape behavior, which falls right at the very bottom of these columns, consistent with a map of kind of the magnitude of the behavior or the coarseness of the behavior onto the location in these columns. So what we have is something that looks like this. We have a transcription factor patterning, and then an age order along the column. And there's actually age order out in the neuropil too. So you can see in a color change experiment, there are green regions and red region, suggesting that what's happening is old cells tend to ramify where other old cells are, and young cells where young cells are, as if you're building circuits in an age order way, where old neurons are connected to other old neurons and young neurons to increasing younger neurons. And physiological properties vary, and importantly, recruitment varies. So I call this the parts catalog approach to building circuits. It's as if, for those of you who built things back in the day, there was a catalog called the small parts catalog where you can get every little tiny tidbit you needed. Amazon bought it and destroyed it, as has happened many times. So it's as if you're building components, and you're building components that can operate over a wide dynamic range. Bigger ones that produce more coarse, faster things, and smaller ones that are producing more slower, more refined things. And that's turns out to be the case. Once you know this exists, you can then ask the question, well, is there any evidence that exists across the phylogeny of vertebrates? And so you look through the literature, and you can find evidence that this is widespread among vertebrate brains by looking at transcription factor literature, morphology literature of neurons at different places in hindbrain, for example. You find that it's in jawless vertebrates. This is birds, mammals, of course. Fish are up here, and amphibians up here. So it seems like it's quite an ancient thing among vertebrates. And most of our work is focused on motor stuff, but what about sensory organization? Jimmy Lau had worked on lateral line system, and he joined the lab to study the lateral line system in zebrafish. And the lateral line system, this is the paper that he published from work done in the lab. The lateral line system, of course, involves these specialized structures called neuromasts that have hair cells that detect water movements along the body. The cells that innervate those hair cells are located in the ganglia, posterior lateral line ganglia up near the head. And it was known at the time that there were variations in the size of the cells in this posterior lateral line ganglia. And there were also variation in how many neuromasts individual cells in this lateral line ganglia innervated. So we actually, before he began the experiments, we made some predictions about what the likely outcome of the experiments would be. And the prediction was that the bigger cells in this ganglia would be earlier born and would innervate more neuromass along the body, thus having a more coarse sensory representation, and the later born cells would have more refined sensory representations. And this just is the figure that really shows the bottom line of the results. So this is the size of the cell axis. This is just the neuromass position along the body. What's important about this is that the size of the circle indicates how many neuromass are innervated by that cell. And the color is from color change experiments he did. The color represents whether it's an older or younger cell. And what you can see from here is that the bigger cells innervate many more neuromass than the younger cells, and I lost my train of thought. They innervate many more neuromass than the other cells and are the early born cells. So the early born cells innervate lots of neuromass, and they're bigger, consistent exactly with the predictions that we made. So this suggests some organizing principles of assembly and function in hindbrain. And so what are these principles? The first is that early neurons and circuits are important for large, fast, coarse movements. Later neurons contribute to more refined, smaller, slower movements. So the circuits kind of differentiate from low resolution to high resolution. And the other principle is that there's a systematic map of function onto birth date, with neuronal circuits formed in age-ordered ways based on the time of differentiation of the cells. So is this just a vertebrate thing? And again, now we can get the answer. That's a question. That's a fair scientific question. We get the answer from the literature for this. And one good place to look is in the escape behavior, which is typically the fastest, most coarse behavior that animals produce. And we can ask, what about the escape behavior across phylogeny? We have a lot of information about escape behaviors across animals because it tends to have big neurons, and so it's easier to study the circuits with big neurons. And so I just want to show you some information about phylogeny, cross phylogeny. We might expect that the escape circuits should be early born big neurons. Now I showed you that that was the case for neurons in zebrafish. But what about, is there any evidence that might be the case for humans? Now this data is not from me, just to be clear. I'll show you some data from me later on. But there is a little bit of suggestive work from humans. This is the movement patterns in utero from ultrasound imaging as a function of weeks gestation. Now in mammals, in humans, the startle response is the equivalent of escape circuit, what happens when the door slams and you jump. It involves much the same circuitry as very short latency pathways as it does in zebrafish. The startle response in humans is one of the earliest behaviors to develop in weeks gestation, suggesting that probably the circuits for that behavior are developing really early. We can't do the recordings in humans, obviously, but at least not yet. What about flies? Fly escape neurons differentiation. So we'll go to this part of this is the phylogeny of all animals, by the way. So we're looking for bigger principles. So fly escape neuron differentiation is earlier. That's the bottom line. So let me walk you through this. This is a paper on the development of giant fiber neurons of Drosophila melanogaster. Now, these folks wanted to understand when the giant fiber neurons are the ones that do an escape behavior in flies, that when you try to swat it, it's hard to swat because it can get away with giant fiber neurons. And they had a line in which the giant neurons were labeled throughout the life of the flies. And that's what's shown here. Now, flies have a rather complex development. They have an embryonic phase, and then they have a larval kind of gross maggot phase, and then they go through several larval stages, and then they have pupal phases as they go through this total metamorphosis into the adult kind of swattable phase with wings and so on. You can swat maggots too, it's just not as pretty. it's kind of gross. So what they wanted to know is when these neurons were born, because it wasn't known. And so they set out to do that by birth dating in flies. And they focused on the larval stages of flies, because neurons are born during the larval stages of flies. And they used BRDU, which is a technique for labeling for birth dating neurons. And they kind of tiled through the larval stages by putting BRDU in and asking if they could get birth date of neurons, if any of these neurons were born during larval phase. And they found no labeling with BRDU of these giant neurons in larval phases. And so that led them to this conclusion, that we must therefore assume that the giant fiber neuron has an embryonic origin. Now this is interesting in the context of these principles. Here's a neuron that's born in the embryo, probably, although it's not definitive, I would say, in this particular case, because they didn't actually figure out when the birth date was. And then it's maintained throughout all of this kind of arcane, dramatic transformations, so that it ultimately produces the escape behavior in the adult fly. This is not just fruit flies. This is a paper studying metamorphosis in flies and other insects. And here they indicate that in both groups of insects, these are the ones with total metamorphosis or partial metamorphosis. The first neurons to be born are large ones, such as motor neurons and interganglionic interneurons. The smaller segmental and local interneurons arise later on, at least in the partial metamorphosis cells. So it seems like it's a broader phenomenon. That's flies and insects. What about nematodes? Does the nematode escape circuit contain some of the earliest born neurons in C. elegans? So nematodes are here. And we know something about nematode escape behavior, not surprisingly. They've been studied a lot. And what happens is, if you touch the nematode here, it withdraws from the touch, and then it locomotes backward to kind of get away from the touch. And that circuit, neurons in that circuit have been identified. And here's a paper, a circuit diagram from a paper. And so I set out to find in the literature when some of these neurons were born, particularly the AVA cells. And the whole lineage of neurons in nematodes is known. Unfortunately, the nomenclature used to delineate the lineage history of cells in nematodes is impenetrable to non-nematode experts. In fact, I think Corey Bargman may be the only one who knows how to read it. But I couldn't figure it out. So I went to the Oracle again. And I asked this question of ChatGPT. Now, knowing that it's ChatGPT is basically not to be believed. It sounds like it knows what it's talking about, but it gets all the facts wrong typically. It's kind of like a politician. And you know how dangerous politicians are nowadays. And so, but actually it didn't do a terrible job on this. It said, yes, the nematode escape circuit does include some of the earliest born neurons. And it even mentions the AVA neurons. They're among the earliest born neurons, interneurons, and C. elegans. Now, needless to say, I did not believe this, okay, for obvious reasons, given what it thought of my work. And so I had to do some more work, and so I consulted a nematode expert, Gunther Hohlepeter at Cornell. He's a biological intelligence, not a chat artificial intelligence BS bot. And he said, I would say AVA cells are born early on in development. So it appears that in nematodes, some of these escape circuits are born early on as well. So that suggests that this principle is broadly present across the phylogeny of the animals. What about the other one? Systematic map of function onto birth date. Let's look at flies first. This is a paper published in 2021, about a decade after our Hindbrain work was published in Zebrafish. It's from Chris Doe's lab at the University of Oregon, and he's a really terrific scientist who's been studying development of fly neurons for a very long time. So you can trust what he says. He's not a ChatGPT. And if you look at the title, it says a developmental framework linking neurogenesis and circuit formation in the Drosophila CNS. You can imagine if you just replace this with zebrafish CNS, you could have a title for our papers that was published in 2011. And it turns out it's very similar to that paper. And I just want to read you something from the abstract. It says, neurons in a hemilineage. Hemilineage is a set of cells that essentially are produced in sequence by cell divisions from a sequentially generated set of progenitor cells. So it's kind of a time series of cells, much like this time series of neurons I showed you in zebrafish. Neurons in a hemilineage share common synaptic targeting within the neuropil. So they're branching in a neuropil, as I said, happens in invertebrates and in larval zebrafish, which is further refined based on neuronal temporal identity. And then they have the beauty of having the EM connectome, and this will be found in zebrafish once people actually have enough easily accessible connectome. That is, connectome analysis shows that hemilineage temporal cohorts share common connectivity. So neurons born at the same time tend to be connected to one another. Now, what about C. elegans? Let's look at C. elegans again. We have to do models because those are where the literature is deepest. This is a paper that just looks at the developmental trajectory of Chinorhabditis. Of course, we know the lineage in nematodes, and we know the connectivity from the EM reconstructions of nematodes. And what this paper asks is, what's the probability that neurons are connected to one another as a function of how far apart they are in the lineage diagram? So this is an example lineage diagram. And so this axis really represents how many nodes are between one neuron that's connected to another neuron. Is it one node or is it many nodes down to a neuron here? And what you find is that the connection probability is a function of the distance apart on the lineage diagram. And this is important. It's both for chemical synapses and for gap junctions, Alberto, and for gap junctions. And this is going to become important, trust me, in the future. Remember, you heard it here. This is going to become important in the future. I have some ideas about this that tell me that this is going to be really important. So there's age-related coupling. That allows signaling molecules to pass between cohorts of neurons in the same circuits. Think about what that might, the implication of that one. So that's what they found. If we look at what they said here, we observed that neurons with certain shared characteristics such as neural process length, birth time cohort, lineage, and bilateral symmetry exhibit a preference for connecting to each other. Okay? Now, that sounds an awful lot like the zebrafish stuff too, I would say. And here's another way of doing it. What you can do is you can just take the pairs of connected neurons and ask how far apart in time are they born relative to one another and make a histogram of the time between the birth dates. And that's what this paper does. So this is the timing in minutes between when the neurons are born, a histogram. And you'll see there's a number of neurons that are born close together in time. And in fact, most connected neurons are born temporally close to each other, with approximately half of them appearing within 15 minutes of one another. So when you're born has a lot to do with who you're connected with. Okay, so I've done flies and nematodes. Most of my work is focused on motor systems, a little bit on sensory systems. But what about other areas of the brain? You know, there's lots of regions that are usually pretty impenetrable. And I want to turn to something that's a hot topic now. It's the fad du jour, and that's cognition. And so I want to say a little bit about areas of the brain that are more integrative. And to do that, especially talk about cognition, you have to talk about mammals because the mammals have defined cognition in a way that applies to mammals. So if you get what I mean. And so I'm going to say a little bit about potential patterning like this in mammals. And the obvious places to study this are in cerebral cortex or perhaps hippocampus. And I'm not a fan of cerebral cortex, as you may have gathered from previous comments. So I'm going to talk about hippocampus. And there was this paper published when I was an assistant professor that made the point that if you look at connections between areas, the highest cortical area is hippocampus. Now, this was troubling to the cerebrocortical people. but it was very satisfying to me. I was very happy about this paper. And the reason these areas are interesting is because there's lots of temporal order in hippocampus and in cortex. We know that neocortex has layers based on timing of differentiation, this inside-out developmental pattern where the oldest cells are here and the new ones are born, migrate past this to form a layering where the younger cells are up here and the oldest ones are down here. That patterning also is present in hippocampus, though it's not as crisply layered as it is in cerebral cortex. Now, hippocampus is thought to be important, very important for representing the context in which things happen. That matters for the formation of memories about things that happen in that context. It's the location of the famous Nobel Prize-winning place cells that are active when an animal is in a particular location, kind of representing roughly where the animal is. So what might we imagine the organization might be like in hippocampus, given this kind of coarse-defined patterning that we see in these other areas? We might expect that neurons, older neurons, deeper layers of hippocampus, might be involved in more coarse aspect of context, and more refined aspects of context, like subtle cues in the environment, might be represented further up in hippocampus. And there's actually quite a number of papers that support this idea. I'm going to show you something from a paper that was just published recently by people in our department, Antonio Fernandez-Ruiz and Ada Oliva. And I had nothing to do with this work. I didn't bias them in any way. This was all done by the time I heard about it. And so I just want to show you what this paper says. Contrary to a common tenet that assumes a random cellular organization of hippocampal spatial and memory representations, we found that pyramidal cells from the same sublayer form functional assemblies. So same sublayer, similar developmental time frame, functional assemblies, reactivated together during sharp wave ripples and have more similar spatial tuning and functional correlates. So they're functionally related to another if they're in the same layer. A larger proportion of active place cells were found among the deep subpopulations, and they were more likely to express place field across different environments, kind of a more coarse representation, deeper, not one very specific place fields, but broader place field. And then this is the most important part. These observations suggest that CA deep and CA superficial cells may play complementary roles in memory formation. Highly active, again, I had nothing to do with this, I didn't see it. Highly active CA1 deep cells may form the backbone of hippocampal spatial and memory representations. On the other hand, CA superficial cells may be recruited after experience and confer higher specificity and discriminability to hippocampal spatial maps and sharp wave associated sequences. So the coarse representation stuff is deeper, and the specificity is in neurons that are younger neurons and more superficial neurons. Again, consistent with these principles of assembly. So some of these principles, if you read the literature, you don't have to do I don't have to do the experiments, I just look at the literature. You find that these are widespread across the phylogeny of animals, suggesting that these principles might be very broad ones of how circuits are assembled and how they work. So the work I talked about was done a long time ago. You might be wondering, what the hell is Fetcho doing now? Actually, I'm kind of wondering that too. And what I've been working on most recently is something related to another aspect of hindbrain that I haven't really talked about. I'm probably way over time, but I don't care. Sorry, Dave. But that's the beauty of being older. What are they going to do, fire me? It's too late, guys. It's too late. I have too much money. It won't matter. Something I haven't told you about hindbrain is that the adult hindbrain doesn't look like the larval hindbrain. In fact, the neurons are not all jammed up in the top of hindbrain, which corresponds to this area here. And instead, the neurons are located in clusters, kind of more nuclear organization in the adult brain. So there's a transformation from the larvae to the adult. And what we thought was going on is that neurons at this stage were forming circuit via very simple rules related to age, and the connections were made down in the neuropil here. And then the neurons are migrating, change their position to different locations in the adult animal. And we could actually see that happen by watching the neurons migrate in vivo in zebrafish, another beauty of a transparent animal. So this is a color change fish in which it's like a 14-hour time lapse. And you'll see neurons migrating out of the old layers and even younger neurons, like in cortex, migrating past older ones to go down more superficially in the bottom of hindbrain. So we can watch the neurons migrate. So we know the cell bodies are migrating. And we thought they were migrating after they formed connections. So Joost Monin actually checked that by tracking neurons over time in 3D reconstructions. Here's a cross-section through hindbrain, and it's at different ages, the same cell over time. At 3 days, 6 days, 17 days. At 3 days, the cell body's up dorsally and has some processes down here, and these are probably likely to be dendritic processes. It's one of those cells that has types that only has commissural axons. And what you can see is that by 6 days, the cell body has changed position. It's down near where these dendritic processes are, and over time it elaborates. So we actually can see that it looks like the connections, at least the branching patterns, are present in connections. We haven't demonstrated exactly that these are connected, so it's not as definitive in that way, but it's very likely. And so it seems very much that there's migration after the connections are formed. Now the problem we faced that prompted some of my latest obsessions is that it was very hard to track neurons in hindbrain after about 17 days post-fertilization because the fish gets much more opaque. And so this prompted me to have another mission, what can I do with my last gasp before I die? And it's the desire to make zebrafish an animal where it can image anywhere in the brain and spinal cord from egg to grave. And to do that, I used an addendum to the tools matter point. Use the most definitive tool, no matter how hard it is, and invent it if needed, or better yet, have your friends invent it. So I moved my lab to Cornell in 2004 because it's the best place, one of the best places in the world for doing in vivo imaging. It has great physicists and engineers. And so I've been collaborating with those people to do 3-photon imaging, which allows deep imaging in the brain of adult animals, mice, and zebrafish. And I just want to show you an example of the latest instrument they built that we've been messing around with. And what this is, I'm not going to describe it all because invite Chris Hsu to come speak at your place. He's the expert on this, and he's a brilliant guy and a very nice person. So what they did was they built a large field of view. The most recent thing they built for us and for them is a large field of view microscope. This images over a 3 millimeter circular field of view, which encompasses much of the zebrafish brain under the objective lens. And it images with 2-photon imaging superficially, and to get deeper, it images with 3-photon at deeper layers. And what this allows you to do is basically image across the brain in one field of view. And so what we're looking at is looking into a Casper fish. And here the neurons are labeled in green with Hijikkamp, and the purple is bone and skin and so on, and myelinated fibers. And so the cerebellum is going to be here, and then we'll see that we're going to focus through from the top of the head down into the deeper into the brain. So this is the cerebellum here. This is the optic tectum. This is the telencephalon. We can basically focus through the telencephalon. This is habenula marni, I think. You can confirm it. And then these are the olfactory bulbs. And you can see the olfactory epithelium even. So you can look across the brain, fairly deep into the brain, in an adult animal. This is important if you want to track changes in the circuitry over time, but it also matters because behaviors emerge from interactions across the brain. And sooner or later, we have to come to grips with those interactions. You have to be able to look across the brain to do that. So that's some of the stuff I've been doing lately. I'm literally doing it myself because I have no people in my lab right now because I'm partially retired, but I love being in the lab, so it's fun. I'm not as good as I once was, though, sadly. So I wanted 2 more things that matter, and I'll be done. Sorry, David. The first one is that brains matter. Brains matter, but some regions matter more than others. And just to give you an example of that, if you have a stroke in your brainstem, the region that I spend a lot of time studying, it really matters because usually you'll be dead if you have a stroke in your brainstem. But that's not true in all areas because some areas matter less. And let me show you a last Z-stack through a brain. This actually is a human brain. It happens to be my brain. And you'll see, I had a stroke about a year ago. And so I'm missing you'll see whiter areas. I'm missing the caudal part of my right frontal cortex, pretty much. And you'll see the white part here. So this is frontal cortex here. This is the caudal part of frontal cortex. This was damaged as a result of this stroke. So some areas matter less than others. I'm here. I gave a talk. And so I exist. And so to hell with ChatGPT and cerebral cortex, as far as I'm concerned. And the last, one more, the most important one, your health matters. So this is the finale, and I'll read it too because it's easier for me. So stay healthy, use your brains while you have them, hint to the older people, to do enduring, definitive work to review principles with strong predictive power. Then tell them to the million people to unmire them from their murk. Rest assured that Zebrafish is the place to find the answers. Thank you.